The overall goal of this simple cell-based bioassay is to detect, quantify, and monitor the activity of members of the vascular endothelial growth factor family of ligands.
This method can answer key questions in vascular biology, virology, cancer and ophthalmology, where VEGF family ligands mediate key processes in these diseases. The main advantage of this assay is it allows evaluation of VEGF family ligands in terms of their capacity to bind and cross-link VEGF receptors without requiring specialised endothelial cells.
Culture the IL-3 secreting cell line WEHI3D by pipetting 5 × 10⁶ cells in the log phase of growth into 4 to 10 T175 flasks containing 50 milliliters of fresh culture medium. Incubate for 7 days, or until the cells have passed their log phase of growth by about 24 to 48 hours. Decant the fluid and cells from the flasks and spin at 1000 Gs for 15 minutes to remove cells and cellular debris. Then remove the top 90% of supernatant and filter it through a 0.22 micron filter unit. Aliquot the conditioned medium into 1 milliliter aliquots for assay preparation, 50 milliliter aliquots for making culture medium to passage of factor-dependent cell lines, and 200 milliliter volumes for long-term storage at -20 degrees Celsius. Store the smaller volumes at -20 degrees Celsius.